Most gardeners make the same mistake every fall. They rake up leaves, bag them, burn them, or send them off the property because they think they're waste. Then in spring, they spend money buying compost, fertilizers, and soil amendments to fix problems those very leaves would have solved. That disconnect is why so many backyard gardens struggle with poor moisture retention, compacted soil, and inconsistent yields year after year. When you remove leaves, you're stripping away the biological engine that builds real soil. You're interrupting the fungal networks, the slow carbon cycling, and the moisture buffering system that nature has refined for millions of years. If you care about growing serious food, not just decent-looking plants, but high-yield, resilient crops, you need to understand the biology that turns dead leaves into garden gold. And if you value practical, field-tested soil knowledge like this, subscribe to the channel now, because this is the foundation of long-term productivity. The real magic starts with fungi, not bacteria. Hot composting has its place, but it's bacterial dominant. Leaves, especially hardwood leaves, feed fungi. Fungi are what break down lignin and complex carbon chains. They don't just rot leaves, they transform them into stable humus. That humus is what gives soil its dark color, sponge-like texture, and long-term fertility. In my raised beds, I've tested side-by-side comparisons for years. Beds fed primarily with hot compost produce fast early growth but dry out quickly in summer. Beds layered with shredded leaves in fall, about 3 inches thick over the soil surface, develop a fungal network that holds moisture deeper and longer. During heat waves, those beds need watering less often and plants show less stress. That's not theory. That's repeated observation. Leaf biology works slowly, and that's the point. When leaves fall, fungi colonize them first. Then springtails, mites, and other microarthropods shred them further. Earthworms pull partially decomposed fragments into the soil. Over months, the carbon stabilizes into humic substances. This process improves cation exchange capacity, meaning your soil holds onto nutrients instead of letting them leach away. If you're working with a small backyard garden, the simplest system is this. Shred leaves lightly with a mower and apply a 2 to 4 inch layer directly on beds after harvest. Water them once to settle. By spring, that layer compresses to about half its thickness and begins integrating naturally, no tilling required. In fact, tilling disrupts the fungal threads you're trying to build. In raised beds, I like a ratio of roughly 2 parts shredded leaves to 1 part existing soil when building new beds. Mix it loosely, not aggressively. The leaves should still be visible. Over the next 6 to 9 months, they'll soften and darken. By the second growing season, the texture difference is obvious. Soil becomes crumbly, not dusty or compacted. Even in hydroponic adjacent systems or container gardening, this biology actually matters more than people admit. Now, I'm not suggesting you dump raw leaves into a deep water culture system, but in cocoa coir or peat-based container mixes, incorporating 10 to 15% well-aged leaf mold by volume dramatically improves moisture regulation and microbial stability. I've tested this in 10-gallon fabric pots with tomatoes. The containers with leaf mold needed watering less frequently and showed steadier nutrient uptake compared to sterile media alone. Roots were thicker and more fibrous, especially in mid-season heat. That's the fungal advantage. It moderates extremes. Most gardening advice ignores soil moisture biology. Gardeners either overwater or underwater because, honestly, they don't actually know what's happening below the surface. Leaves and leaf mold increase water holding capacity, but they also improve drainage structure. That balance is biological, not mechanical. This is where I rely on a simple tool to take out the guesswork. I use a 4-in-1 soil moisture meter that measures soil moisture, pH, temperature, and sunlight intensity. The link is in the description if you want to check the exact model I use. It's not about gadgets, it's about data. When you're layering leaves or integrating leaf mold, you need to know if your soil is staying evenly moist or, becoming anaerobic. For example, fungal activity thrives in moist but oxygenated conditions. If your moisture reading is consistently maxed out, you've overdone it and risk compaction. If the soil temperature drops too low and moisture disappears, fungal breakdown slows dramatically. In my beds, I aim for moderate moisture, not saturated, and soil temperatures that stay biologically active through fall. That feedback loop really speeds up the transition from leaf litter to stable humus. Let's talk about some common myths that honestly hold gardeners back. One myth is that leaves steal nitrogen. Fresh leaves can, temporarily tie up nitrogen at the surface, but that's really only an issue if you're planting directly into thick, undecomposed layers without giving time for microbial adjustment. The solution isn't avoiding leaves, it's timing. Apply them in fall, or at least several weeks before planting. Now, if you're in a short-season climate and applying in spring, a light dusting of composted manure beneath the leaf layer balances things easily. Another myth is that all leaves are equal. They're not. Thick, waxy leaves like magnolia break down slower than, say, thin maple leaves. That's not bad, it just means you adjust your expectations. Diversity is ideal. Mixed leaf species create more complex fungal communities, which, in turn, improves overall soil resilience. If you want to accelerate the process without destroying fungal dominance, shred the leaves and keep them, just slightly damp. I've built simple leaf bins using wire fencing, about 4 feet wide and 3 feet tall. Fill them up, water lightly, and let them sit. Turn once or twice a year at most. Within 9 to 12 months, you'll have dark, earthy-smelling leaf mold, not hot compost, not steaming piles, just stable, fungal-rich material. This is where dead leaves become true garden gold. When leaf mold is mature, it feels kind of like a wrung-out sponge. It crumbles easily and smells like forest soil. Apply 1 to 2 inches as a top dressing around heavy feeders like tomatoes, squash, and brassicas. In fruit tree guilds, spread it in a 3-foot diameter ring under the canopy. Over time, the soil structure beneath improves dramatically. In one of my older raised beds, after 3 consecutive years of leaf layering, I actually stopped adding purchased compost entirely. The soil maintained fertility through cover cropping and leaf applications alone. Yield stayed consistent, watering frequency decreased. Pest pressure even seemed lower, likely because healthier plants attract fewer opportunistic issues. The forgotten biology is patience paired with observation. Leaves are not a quick fix. They're infrastructure. They build the carbon backbone of your soil. They feed fungi that connect plant roots through mycorrhizal networks. They create aggregation that prevents erosion and compaction. None of that happens in a 2-week hot pile. If you're serious about results, stop thinking season to season. Think in cycles. Every fall leaf drop is an opportunity to invest in next year's fertility. Measure moisture, monitor pH, observe root health, adjust ratios. That's how you refine the system. And here's the bigger picture. When you keep leaves on your property and cycle them through your beds, you close the loop. You reduce dependency on outside inputs. You build soil that improves instead of degrades. That's the difference between gardening and soil stewardship. If this kind of grounded, experience-based approach to growing food resonates with you, subscribe to the channel. Share this video with another gardener who still bags up their leaves every fall. The more we understand the biology beneath our feet, the better food we grow above it. Dead leaves aren't waste. They're the raw material for the most powerful soil system you can build.